So till now we have discussed about different mechanistic aspect of homologous recombination, but we need to discuss about few important clinical relevance of homologous recombination. Now, homologous recombination, because we already know that it is linked to double-strand break repair. So it is, of course, very important in terms of maintenance of genomic instability, and also it is important in diversification of different pathogenic organism. So here in this particular short segment, I would like to discuss about different clinical aspects of homologous recombination. Now, there are at least there are four different clinical aspects that I would like to touch upon. The first one is the dissemination of antimicrobial resistance in bacteria. Now, this should be a very long discussion, so I will not get into intricate details of it. But since we all know that horizontal gene transfer is mediated by events like conjugation, transduction, and transformation, whereby some bacteria can acquire foreign DNA, and that foreign DNA, if it contains some antimicrobial resistance genes, particularly genes that can confer resistance against some bacteria, so against some drug antibiotic, the bacteria would become resistant to the antibiotic, and thereby it would show antimicrobial resistance. If multiple such genes are acquired by some bacteria, and those genes are following homologous recombination, those genes are stabilized on the bacterial genome, thereby the bacteria would acquire multi-drug resistance. Both antibiotic resistance as well as multi-drug resistance thereby can be linked with the event of homologous recombination. Although site-specific recombination and transposition, these are also involved in dissemination of antimicrobial resistance. But as I mentioned, I will not get into the mechanistic intricacies of these processes. The second important aspect of clinical relevance is pathogenicity and antigenic variation. Now, again, just like antimicrobial resistance, site-specific recombination as well as your transposition might contribute to such variations, pathogenicity, acquisition, and antigenic variation. But homologous recombination is also involved in such events, particularly in one parasite that we would discuss. It's Trypanosoma brucei, where the surface glycoprotein expression is dependent on homologous recombination. So we will discuss that in detail. Then in terms of drug resistance for cancer as well as for some parasite. So here we would discuss drug resistance in Leishmania, how homologous recombination is linked to drug resistance in Leishmania, another protozoan parasite. And lastly, we will discuss about some event which is known as homologous recombination deficiency, where some cancer cell can become sensitive to some drugs due to deficiency of homologous recombination. So those we will discuss. So let us start with Trypanosoma brucei, the parasite that causes sleeping sickness. This is one of the major major concern in terms of parasitic diseases after malaria and leishmania. So this parasite causes African trypanosomiasis, or which is also known as sleeping sickness, and this parasite is a vector-borne disease. It is transmitted by tsetse flies, and it can bite human, and thereby the parasite gets into the human system, where it can proliferate, and it can differentiate into several forms like procyclic form, metacyclic form, stumpy form. All of those forms it can change, and it can affect our nervous system and other systems to elicit the pathogenicity. Now, at the cellular level, the parasite is coated with a protein, a glycoprotein that is known as variable surface glycoprotein or VSG. VSG can play a very important role in pathogenicity of the parasite. Particularly, it can inactivate complements. Complement system, we all know it is active in lysing the cells, so lysing the invading cells. So complement inactivation helps the parasite to grow inside our system. Then it can generate autoantibody, which is linked to the pathophysiology of the disease. So generation of autoantibody by molecular mimicry. Then VSG can trigger pro-inflammatory cytokine production like TNF alpha and others. So which is also linked to the neuroinflammation and other kind of pathogenic condition. And also the most important one is the antigenic variation, where the parasite expresses different types of variable surface glycoproteins, and thereby the parasite is circumventing our antibodies and immune system per se. So this is a very important strategy for Trypanosoma brucei to infect us. Now, BSG is encoded by several genes in the genome, and individual trypanosomes can harbor different sets of variable surface glycoproteins. But the expression would be on only one copy of those variable surface glycoproteins. So far, we have identified thousands of such VSGs, and the VSG genes has been found to be scattered among the chromosomal materials of the parasite, and thereby there can be multiple VSG genes located in multiple locations on a chromosome. But the VSG that is expressed, these are called expression linked copy. So it is nearby some promoter or some transcriptional start site. Actually, transcriptional start site should be the correct word. And then if a copy linked to a specific transcriptional start site, that would only be expressed. Now in this parasite, the due to homologous recombination, such copies can be changed. Some other VSG gene can replace the expression linked copies, and thereby a new VSG gene will be expressed. So therefore this particular process, which is similar to gene conversion in mating type switching in yeast, here a specific copy which we call the basic copies or the VSG repertoire, any one copy of VSG repertoire can combine with the expression site like you can see here, and thereby it can form a expression of another copy of VSG gene. Thereby the surface glycoprotein can vary. So you can see here this red patch is the expression linked copy, and here you have the VSG cluster, different types of VSG genes, and recombination event can occur, and this event can lead to formation of new types of VSG genes, or this recombination event can also lead to replacement of the pre-existing VSG gene with some other one. So therefore this kind of gene replacement or gene conversion might be the gene conversion leads to expression of different types of VSG gene in trypanosoma, and thereby the surface glycoprotein variation is accomplished in the parasite. The next topic that we would like to discuss is genetic recombination in leishmania, which can lead to drug resistance. Now, leishmania genome and genome of Cyanidoplasida parasites per se, it contains immense number of repetitive sequences. And as we discussed before, that recombination between direct and indirect repeats can have its own implication. So in leishmania, recombination between such direct repeats can lead to the generation of circular amplicons. So recombination between such two direct repeats can lead to formation of circular amplicons. Then in leishmania, there during recombination, there can be unequal exchange of DNA between sister chromatids, and these unequal exchange of sister chromatid might lead to formation of intrachromosomal tandem duplication of specific DNA sequences. These are called linear amplicons. So such intrachromosomal tandem duplication also increases copy number of the gene. The circular amplicon generated by recombination between two direct repeats can also amplify, and therefore the number of copies that are harbored, gene copies that are harbored within this amplicon, will ultimately grow in number. Now, if there is some drug resistance gene like MRP8, one of the efflux pump, drug efflux pump, or there is some drug modifier within this particular region, then what would happen? Then these uh, these uh, these amplification, tandem or bicircular amplicon, would lead to increase in the copy number of the resistance factor. So more efficiently the parasite will be able to drive out the drugs or resist the drugs. So thereby homologous recombination in this parasite actually assists the parasite to attain resistance against specific drugs. This is also clinically very important, and this is also experimentally proven, as well as it was found out that in some clinical isolates such amplicons are present. So which means that during drug treatment the recombination event might be triggered to enhance the copy number of the resistance genes. So this is also very important. Now, last topic that I would like to discuss now is homologous recombination deficiency. It's a phenotype that is characterized by the inability of the cell to effectively repair the DNA double-strand breaks. Now, we know when some DNA double-strand break is induced by some damaging agent or some other factor, normally there is a signaling cascade that is triggered, ATM kinase mediated signaling cascade, which subsequently activates DSBR, and therefore the damage is repaired. Now, if this whole system somewhere is somehow impaired due to lack of a good RAT51 or good copies of these homologous recombination machinery genes, there might be homologous recombination deficiency. And this homologous recombination deficiency would actually promote tumorigenesis, because this deficiency would mean immense genomic instability, and therefore it would lead to tumorigenesis. We have to remember that double strand break repair is one of the most accurate repair mechanisms. The sequence is not changed, or the whole arrangement of the genes is not actually changed, but our non-homologous end joining or other kind of repair mechanisms, those are not really that accurate. So therefore disruption of such an accurate repair mechanism leads to genomic instability and enhances tumorigenesis potential. In some cancers, particularly like ovarian cancer and some other cancers, it has been observed that some cells or some tumors can become deficient in homologous recombination. Now, it has one advantage clinically. The advantage is when some cells become deficient in homologous recombination, that would mean that they would become vulnerable to or susceptible to uh, drugs that can inhibit PARP, um, ADP ribose polymerase, PARP, which is again a repair protein, and also it would some the drugs, particularly drugs like the platinum-based chemotherapeutic drugs, it would become more susceptible to those drugs. So in a way, this deficiency on one hand can trigger tumorigenesis. On the other hand, such deficiency can actually make the cancer cell susceptible to drugs like PARP1 blocker or some other drugs, leading to cell death. Okay, so therefore homologous recombination deficiency is a very important uh, mm, uh, phenomena, and now people are trying to detect whether tumor cell is indeed deficient in homologous recombination, so that that can be cured by such drugs, PARP inhibitor or platinum drugs. So this way, homologous recombination is also associated with precision therapeutics or precision medicine. So precision medicine means prescribing a drug for a particular condition. So you can prescribe such drugs for homologous recombination deficiency. So if you can detect homologous recombination deficiency, it would be easier to treat the cancer. Therefore, recombination or homologous recombination is very significant clinically, and we must remember that there are many other clinical relevances of homologous recombination. I just mentioned a few because I think that this kind of clinical relevance must be discussed along with the typical mechanism of the processes. So I have attempted to provide a broader spectrum of homologous recombination through this series of discussions. I hope I was quite clear in terms of stating different facts and explaining different experiments as well as different mechanisms. Thank you for your patience, and thank you for your attention. Goodbye.